and welcome back to Vet Reviews with Doc Athena. Welcome to Vet Review with Doc Athena. We're back to basics. Definition of terms, Part 1, erythrocytes. So, how much do you remember? Let's find out. Let's define erythrocytes first, okay? Erythrocytes are mature red blood cells, or RBC, and a complete evaluation includes PCV, TEC, morphology, parasites, hemoglobin, MCV, MCH, and MCHC. So, first question. What hormone stimulates the increase in production and release of erythrocytes by normal bone marrow? 2, 1, 10. Answer, erythropoietin. Very good for those who were able to get the correct answer, and those for not, well, better luck next time. There's still a lot of questions coming on our way before we end this video. Let's go back to our lesson. All right, so what is PCV? This is packed cell volume. This is also known as microhematocrit. That's why the little tube that we use in the laboratory to collect the blood and run in the centrifuge is called microhematocrit tube. And this determines the percentage of blood composed of erythrocytes. Remember, PCV is for percentage, and let's differentiate that with TEC. Okay, so TEC is the total erythrocyte count. This reflects the total number of RBC in the circulating blood, and this is used to determine the functional state of erythron. Now, this does not indicate the oxygen or oxygen carrying capacity of the RBC. Okay, so for PCV, it's percentage, and for TEC, it's the total number. Okay. Now let's proceed to morphology. Morphology reporting includes degree of polychromasia, anisocytosis, poiculocytosis, rouleau formation, parasites, and inclusion bodies. We will discuss this one by one, but first, let's have our second question. What is poiculocytosis? You have 5 seconds to give me your answer. 4, 3, 2, 1, set. Answer? Poiculocytosis is the variation in erythrocyte shape. Okay, remember that. Poiculo, shape. Okay, cytosis pertains to the changes or abnormalities with the cell. Okay, so poiculocytosis is the variation in erythrocyte shape. Okay. Now, let's talk about the different types of poiculocytosis. First is the acanthocytes. All right, acanthocytes have variable length projections. Okay, normally the red blood cell is biconcave in shape, normally, okay? All right, so you have a photo here on the right. Disclaimer, this is not mine, and let's recognize the source, okay? This was from Usman, Nimji, and Hagestorm, MD, and it was taken from Wikipedia, all right? So, acanthocytes, variable length projections. Okay, so there are many projections in the RBC with different lengths, okay? And usually, this indicates hepatic diseases, CIVD. What is CIVD? This is disseminated intravascular coagulation and in cases of hemangiosarcoma, which is one type of neoplasm. All right, next type of poiculocytosis is echinocytes. And this time, the projections are uniform. Here's the echinocyte, all right? And this is the acanthocyte. All right, so you see the projections. For echinocyte, the projections are almost uniform, whereas for acanthocyte or spur cell, the projections are of various lengths, okay? Now let's proceed to Dacrosite. Do we have Dacrosite here? Yes, here, the third drop cell, okay? And in Liama, these are usually associated with iron deficiency, and for calves, this is associated with phenylhydrazine-induced hemolytic anemia. Okay, schizocyte. I think here we have schizocyte here. These are fragmented erythrocytes, and this is usually associated with intravascular trauma. All right. How about spherocytes? Spherocyte is the RBC looks more of spherical rather than biconcave. And usually this is associated when there is a lost portion of the membrane. So these are the common types of poiculocytosis. Now let's proceed to the third question. All right, what is anisocytosis? I'll give you 5 seconds. What is anisocytosis? 2, 1, 0. Anisocytosis is the variation in erythrocyte size. For poiculocytosis, this is the variation in erythrocyte shape. Whereas for anisocytosis, this is the variation in erythrocyte size. Okay? Poiculo, shape, aniso, size. Remember that. All right. Now, let's talk about the changes in the sizes of our erythrocytes. We have rule formation. Okay, rule formation is the stacking of erythrocytes like coins or a grape-like cluster. And this happens usually due to the coating of antibodies common in cases of immune-mediated hemolytic anemia. And in order to verify this, we can add 1 drop of saline in less than 1 drop of blood. Now next, basophilic stippling. These are small basophilic dots in erythrocyte due to aggregate ribosomes. And usually this is common in cases of regenerative anemia and lead toxicity. Now there's Heinz bodies. Heinz bodies occur when? Usually when there is onion poisoning, right, with the dogs. And you can see this under the microscope as a small circular structure within or protruding from erythrocyte. This is due to the denatured hemoglobin precipitate as a result of oxidative damage, usually because of GSH deficient. Okay, another inclusion body is the Howell-Jolly bodies, okay? And these are small, round basophilic remnants. This is common in cases of regenerative anemia and splenic dysfunction. Next is the Nrbcs, or nucleated red blood cells. And these are actually immature erythrocytes. All right. These Nrbcs usually show up when there is lead toxicity or regenerative response to anemia. Now, what is polychromasia versus hypochromasia? Polychromasia is a color variation due to the presence of reticulocytes, which stain bluish due to residual cytoplasmic DNA. Okay, and this is another sign of regenerative anemia in ruminants. On the other hand, hypochromasia is increased central pallor in blood film due to low hemoglobin content, a sign of iron or copper deficiency in ruminants. However, in ruminants, this is difficult to recognize due to their small size of erythrocyte. Okay, this is actually from my thesis. This is my undergraduate thesis when I was in college, all right? And it's all in here that I'm sharing with you. It's just basically a review. So polychromasia is when there's increased, okay, variation in colors. It appears bluish because of the reticulocytes, whereas hypochromasia, there is an increased central pallor. Okay, so that's a major difference of the two. Now let's proceed. Okay, now let's proceed to the parasites of erythrocytes. Usually there's anaplasma species, mycoplasma, or epitherozoon species. There's also babesia species, theileria species, and trypanosoma species. Okay, we will discuss these blood parasites in more detail on a separate video, all right? Now question, another question. Blank is the decrease in erythrocyte number or oxygen carrying capacity of the blood. What is this? I'll give you 5 seconds. 4, 3, 2, 1, 0. Okay, this is anemia. Anemia is the decrease in erythrocyte number or oxygen carrying capacity of the blood. All right. Okay, so now if hemoglobin is known, corpuscular values can be computed. Okay, and this is important to know the type of anemia. Now, we have 3 values, MCV, MCH, and MCHC. MCV is the mean corpuscular volume, and this is the average cell size, okay, size, and it is expressed in femtoliter or FL. Normal size is called normocytosis. Increase in sizes would be macrocytosis, and decrease in size would be microcytosis. Okay, whereas MCH is a mean corpuscular hemoglobin. This is more useful because it expresses the average cell hemoglobin content, and this is expressed in pictograms or PG. All right. Whereas, mean corpuscular hemoglobin concentration. Sorry, I think this one is the most useful of the 3. The mean corpuscular hemoglobin concentration, or MCHC, is the average cell hemoglobin concentration, and it is expressed in percent. Okay? If it is normal, it is called normochromic. If it is increased, it is hyperchromic. And if it is decreased, it is hypochromic. All right? If we're talking about size, this is normocytosis, macrocytosis, or microcytosis. But if we are talking about the hemoglobin concentration, this is expressed in percentage. It is usually described as normochromic, hyperchromic, or hypochromic. All right? So that's it for our definition of terms part 1, erythrocytes, for our Back to Basics in Pathology. This is Doc Athena for Vet Review. Thank you. Bye. I hope you remembered something from this vet review with Doc Athena.